gram-negative aerobic or microphilic rods and cocaine. Group 4 gram-negative aerobic or microphilous rods and cocaine. Pseudomonas, genus Pseudomonas, Xanthomonas, Juglia, Gluconobacteria. Correct, right? G negative, slightly covered, motile bipolar flagella, oxidase positive, catalase positive aerobic, respiratory metabolism plus never fermentative. Growth requirement are simple: short soil and water. Genus Pseudomonas. Pseudo means false, monas means unit. False species of monas, an early generic name of protozoan. Many species of Pseudomonas produce fluorescent discernible pigments of a greenish yellow or yellow-green color, black or blue. Optimum growth temperature 25 to 30 degrees Celsius. Nutritionally versatile, grow well on solid media. Presence of the organism in milk and milk product is highly objectionable because produce heat stable protease and lipase even at low temperature. Versatile spoilage agent attacking fat and protein include pathogens which cause milk-borne illness. They have very little fermentative activity on carbohydrate. Some of these produce phosphatase, and if they grow in pasteurized milk, they cause false positive test. Pseudomonas fluorescens produce water-soluble fluorescence greenish brown pigment known as pyoverdine. Optimum growth temperature 25 to 30 degrees Celsius but grow at 5 degrees Celsius. Causes bitterness and lipolysine in refrigerator foods by elaborating proteinases and lipases and gelatin in UST product. This also produce phospholipid and glycosides but cause no coagulation of litmus milk. Pseudomonas fragi rarely produce pigments, sometimes diffusible brown pigment. Grow at 5 degrees Celsius. Optimum growth temperature 20 to 25 degrees Celsius. It is mostly lipophilic and rarely zeolitic. Produce fruit-like odor resembling apple or strawberries. Compounds responsible are ethyl butyrate and ethyl hexonate. Pseudomonas aeruginosa produce greenish blue pigment, pyocyanin and pyoverdine and non-carotenoid pigment. Do not grow at 5 degrees Celsius but brown at 41 to 42 degrees Celsius. Optimum growth temperature 37 degrees Celsius. Opportunistic pathogen causes mastitis. Genus Xanthomonas maltophila, previously known as Pseudomonas maltophila. Differentiating characters and Pseudomonas are produce yellow non-diffusible cell-bound pigment. Inability or reduced nitrate required methionine as growth factor. No growth at 4 degrees Celsius and 42 degrees Celsius. May grow at 41 degrees Celsius. Optimum temperature 30 to 35 degrees Celsius. Utilization of citrate negative. Genus Alteromonas. Alteromonas putrefaciens, formerly known as Pseudomonas putrefaciens, produce a non-diffusible pink or reddish-brown pigment. Produce H2S or king iron agar, rod-shaped. Optimum growth temperature 20 to 25 degrees Celsius. Major proteolytic and less lipolytic in nature. Cause surface strain in bitter and also cheesy. Putride flavor defect. Source water and soil. Genus Echinacea. alkaligenous. Rod or cocaine, motile, obligate, aerobic, psychotropes, and some are thermodiuric. Causes droopiness in milk and milk products. Alkagenous, viscous, alkagenous tolerance. Genus Brucella. Genus Brucella. Gram-negative dot short ellipsoidal small circular convex colonies. Optimum growth temperature 37 degrees Celsius. Non-motile growth may favored with increased CO2 tension. Characteristics: Brucella abortus, Brucella suis, Brucella melitensis. Growth in presence: methyl violate positive, negative, positive. Urease negative, positive, negative. H2S production negative, negative, positive. Abortion: cow, swine, goat, and sheep. Group 5 facultative anaerobic gram-negative rod. Group 5 facultative anaerobic gram-negative rod. General characteristic: straight rod, motile by peritrichous flagella or non-motile. Aerobic and facultative anaerobic grow well at 37 degrees Celsius and often produce acid and gas. Generally referred to as coliform, which may be defined as gram-negative, oxidase negative non-sporing rod, which can grow aerobically or facultatively anaerobic in the presence of bile salt and able to produce acid and gas within 48 hours at 37 degrees Celsius. Gram-negative, oxidase negative non-sporing rod. Enterobacteriaceae. Genus Escherichia. Enterobacter, Salmonella, Yersinia, Citrobacter. Characteristics: gram-negative, short and straight rod, motile by peritrichous flagella or non-motile, may be capsulated aerobic or facultative anaerobic. Chemoorganotrophs ferment glucose producing acid or acid and gas, mainly catalase positive, oxidase negative, inhabitant of the intestine of man and animal, and some may act as pathogens or saprophytic. Coliform. Coliform may be defined as gram-negative, oxidase negative, non-sporing forming rod, which can grow aerobically or facultatively anaerobic in the presence of bile salt or surface active agent with similar growth inhibitory properties and which are to ferment lactose with production of acid and gas within in 48 hours at 37 degrees Celsius. Genus Escherichia. Genus Escherichia. E. coli, E. blati, glucose and carbohydrate, pyruvate, lactic acid, acetic acid, and formic acid. Formic acid, CO2 and H2O, and CO2 and H2. Litmus milk get coagulated with rapid acid production and gas. Citric acid and its salt are not utilized as sole source of carbon. OGT 30 to 37 degrees Celsius but grows at below 10 and above 45 degrees Celsius. About 30% of the acid produced by E. coli is lactic acid, and so they are also called pseudolactic acid bacteria. The type of acid produced by E. coli depend on sugar fermented and nature of end source. Significance: their presence in the food is indicative of fecal contamination. They are protein food spoilage organisms. They produce gas in the dairy products. They produce unclean flavor in the dairy products. Some strains produce droopiness in the dairy product. Their presence in pasteurized food indicates unhygienic production at the plant. Some strains produce enterotoxin and some are enteropathogenic in nature. Motility: motile, non-motile. CO2 H2 1 is equal to 1 2 is equal to 1. Origin: fecal, non-fecal. IMV-ICT test: indole positive, negative. Methyl red positive, negative. Vegas proscore negative, positive. Citrate negative, positive. Result of IMV-IC test of E. coli. Indole test, methyl red test. Vegas precursor test, citrate utilization test, genus Enterobacter, Enterobacter erogens, E. Enterobacter cloacae, fecal or non-fecal, ferments glucose by means of butanidol forming fermentation or produce acetyl methyl carbinol to give positive Vogus Proscott test. Encapsulated variant cause ropiness. Genus Salmonella. Salmonella is named after D.E. Salmon, USA. Gram-negative aerobic and facultative anaerobic with optimum growth temperature at 37 degrees Celsius, mostly motile by Peritrichus flagella. They produce acid and gas from carbohydrates such as glucose, maltose, sorbitol, etc. except Salmonella typhi. It does not ferment lactose, positive for methyl red, negative for indole and Vogus precursor, but variable in utilization of citrate. Source: direct or indirect fecal contamination. Cows suffering from salmonellosis. They are designated into by Kofferman's wide serotypes such as oosomatic as flagella and IV virulence antigens. It is an important group of organisms because of its ability to produce a variety of food infections and illness. It produces endotoxin 12 to 30 hours may elapse after injection of food contaminated with these organisms, and the onset of symptoms are due to the elaboration of endotoxin by the growth of the organism. The following illnesses are associated: typhoid, Salmonella typhi, paratyphoid, Salmonella typhi A, typhi B, typhi C. Genus Yersinia. The important organisms are Yersinia enterocolitica, Yersinia pestis, and Yersinia pseudotuberculosis. Yersinia enterocolitica enter milk by contamination through feces, urine, and insects. They grow between 2 to 45 degrees Celsius with OTG of 30 degrees Celsius. Yersinia enterocolitica. All the organisms of the genus are pathogenic to human and or animal or opportunistic potential pathogen. Genus Serratia marcescens produce red pigment known as prodigiocin. Genus Vibrio cholerae cause cholera in human. Group 9 Rickettsia and Chlamydias. Group 9 Rickettsia and Chlamydias. Coxiella. The genus Coxiella is the only important genus of interest to the dairy microbiologist. Coxiella burnetii is a rickettsial organism. This is the causative organism of Q fever. The cells are short rod and exhibit a filtrable phase in their development life cycle. C. brunetii has a high degree of resistance to chemical and physical agents, including desiccation, and will survive for long periods in tissue or feces of infected and in wool, etc. C. brunetii is a rickettsial organism like intracellular parasite which will localize and proliferate within the cell vacuoles. Tick and ruminants, including dairy cattle, sheep, and goat, are common asymptomatic carriers of C. brunetii, with most human cases being traced to dairy workers, farmers, and meat factory employees who work close contact with animals. Injection of raw milk has been linked to Q fever in the United States and England. Heating raw milk at 62.8 degrees Celsius for 30 minutes or 71.1 degrees Celsius for 15 seconds is sufficient to completely destroyed serenity. Infected animal which may apparently be healthy may excrete the organism in their milk and large number are released during parturition. The aerosol route may infect man. The organism also show high resistance to chemical and physical agent and desiccation. It is resistant to 0.5% formalin, 1% phenol and can withstand the heat treatment of 60 degrees Celsius for 1 hour and 61.7 degrees Celsius for 30 minutes. Group 70 gram positive cocci. Group 70 gram positive cocci. Genus Micrococcus, Staphylococcus. These two genus consist of gram-positive spherical cells. They are non-motile, aerobic or facultative anaerobic, chemoorganotropes and catalase positive. Growth anaerobically negative positive, carbohydrate oxidative fermentative, lysotrophin sensitive negative positive, ability to ferment glucose anaerobically negative positive. Aerobic coagulase negative mesophilic form tetrad. Some of the organisms produce yellow organ red pigment. Lactose is non-formed OTG 25 degrees Celsius. Many species are heat resistant and service 63 degrees Celsius or 30 minutes, but the true micrococci are not resistant to pasteurization. They are found in lactiferous duct of mammary gland and present in milk obtained from udder under sterile condition and hence considered as normal microflora of milk. Contamination equipment is the main source of micrococci in milk. They are responsible for thickening of sweetened condensed milk and cause thermoduric outbreak in pasteurizing plant. Example: M. varians, M. luteals, genus Staphylococcus. They are cocci and smaller than micrococci. Occur in grape-like cluster. Non-motile, may produce organ or yellow pigment. Staphylococcus mesophilic OTG 37 degrees Celsius. Ferment a variety of carbohydrate and resistant up to 10% of salt concentration. They are capable of producing several toxins like hemolysin, fibrinolysin, leukocidin, enterocotoxin, and thermostable nuclease, etc. Hemolysin are the substance that liberated hemoglobin from RBC. Beta hemolysis produce clear colorless zone around colonies. Hemoglobin destroyed to produce colorless compound. Alpha hemolysis convert hemoglobin to methanoglobin which produce greenish zone, short, skin, nasal and mucous membrane. Staphylococcus aureus. Staphylococcus aureus. Facultative anaerobe growth at 50 and 45 degrees Celsius and is mesophilic. The colony are with yellowish staining lemon and organ yellow. Endospore forming gram-positive rod and cocci. Endospore forming gram-positive rod and cocci. Genus Bacillus. Bacillus means little stick, gram-positive large rod. Aerobic saprophytic soil bacteria may occur in single pair or chain. Aerobic spore formation source air, water, soil, feed and fodder. Isolated by heating raw milk to 80 degrees Celsius or 10 minutes before plating. Large and rough colony are formed acid-sensitive potent spoilage organisms. They may psychotrophs, mesophilic, thermophilic, catalase positive. Spore ellipsoidal to cylindrical in shape, occasionally bulge sporangia. Bacillus cereus. Bacillus cereus do not grow at 5 degrees Celsius but grow at 20 to 35 degrees Celsius. This is important organism because cause food poisoning by produce enterotoxin when the numbers exceed 10 to the power 6 per gram. Produce bitty cream defect, broken cream by the action of lecithinase enzyme, extracellular phospholipase. Produce sweet curdling by coagulating milk at lowered acidity by producing rennet-like enzyme. Bacillus subtilis growth between 20 to 45 degrees Celsius. This is important organism because cause ropiness or slimness in raw pasteurized milk, causes spoilage of UST concentrate and or canned milk product, causes sweet curdling by coagulating milk at lower acidity by producing rennet-like enzyme, produce leaven extracellularly from sucrose. Extracellular enzymes, including those that degrade protein, acid, polysaccharide, or plant tissue, are produced. Polypeptide antibiotic subtilin are produced. Bacillus licheniformis, Bellus licheniformis. This is important organism because causes spoilage or UST concentrate or canned milk product produce leaven extracellularly from sucrose cause rapidness or slimness in raw pasteurized milk. Bacillus coagulans, these cause spoilage of UST concentrate or canned milk product. Bacillus coagulans and B. stereothermophilus grow between 45 to 65 degrees Celsius. This is important because it is obligatory thermophilic, cause flat sort spoilage of canned or UST milk product. Used in the detection of antibiotic in the milk. Antibiotic acid test. Genus Clostridium. These are the anaerobic organisms and spore former gram-positive rod with 1 to 4 m in length, motile catalase negative. They may be mesophilic or thermophilic. The important source of this organism is soil, intestinal tract of animal. It may become established as contaminant on equipment. They gain entry into milk via feces, soil, feed, and especially silage. Some are pathogenic organisms such as C. proliferans, Clostridium botulinum, Clostridium butyricum, and Clostridium typebutyricum are thermophilic. Their fermentation and products include acetic acid, butyric acid, butanol, isopropyl alcohol, acetone, H2, and CO2. Clostridium perfringens cause bovine mastitis in cattle and cause neurological disorder in humans. It produces profuse gas and break the coagulum of milk causing stromy fermentation. Clostridium trobutyricum cause late blowing condition in cheese. Clostridium butyricum cause late blowing condition in cheese after 1 to 2 months of manufacture. Clostridium sporinges develop rancidity of immaterial cheese. Staphylococcus bunch of grape-like appearance. Hemolysin alpha, beta, gamma, and delta are produced sensitive to novobiocin. Most of the strains produce enterotoxin and exotoxin, which are responsible for food intoxication. 5 different enterotoxins are reported. They are A, B, C, D, and E, but enterotoxin A are more common. They are heat stable and produce symptoms in 2 to 6 hours after injection of contaminated milk. Medium on Bayer-Parker's medium, they produce black semi-convex colonies surrounded by a clear zone. In the clear zone, a fine black precipitate may appear. Example zone of opalescence. The pathogenic nature is confirmed with a positive coagulase and thermonuclease test. It produces bacteriocin via staphylococcus or micrococci, which are bacteriostatic or cidal to bacteria and to other staphylococci. Other organisms of staphylococcus found in milk are as epidermis, as capri, as Hylicus, genus Streptococcus, genus Streptococcus. Strepto means flexible, a pliable length of cocci similar to necklace. Gram-positive, spherical or ovoid and occurs in pair or chain. Fastidious organism, facultative anaerobic. Sherman 1937 divided Staphylococcus into four groups based on serological active group, species specific polysaccharide C substance into pyogenic viridian Enterococcus lactic group. Pyogenic group. Pyogenic group. Streptococcus pyogenes belong to Lancet A. It is a pathogenic organism and causes septic sore throat, scarlet fever. The habit is upper respiratory tract, skin lesion, and inflammatory exudate. It produces beta hemolysis and resists phagocytosis. It produces erythrogenic toxin. It causes acute mastitis in animals. CAM test is positive. Streptococcus agalacticae. It belongs to Lancefield beta and it is alpha weakly beta gamma hemolysin. It is similar to S. pyogenes and causes mastitis in cow. It causes meningitis and pneumonia in human. CAM test is negative. Streptococcus dysgalacticae. Lancet-filled group C causes bovine mastitis. CAMP test is negative. Viridans group Streptococcus uberis. They cause winter mastitis in cow. Serological heterogeneous. Non-hemolytic CAMP test is negative. Streptococcus thermophilus. Streptococcus thermophilus. This is now known as Streptococcus salivarius species thermophilus. It has no group-specific antigen. Growth at 45 degrees Celsius but not at 10 and 53 degrees Celsius. OTG is 40 to 45 degrees Celsius. It is thermophilic starter culture used for the preparation of yogurt, Swiss cheese. This is used for antimicrobial agent as example sulfadiazine. This can be used in microcapsulated from when grown in milk with added amino acid along with medium and also used in hypocaloric diet for treatment of obesity. On yogurt, lactic agar shows a small white colony with clear zone where at Lactobacillus delbrueckii species bulgaricus produce large white colony surrounded by cloudy zone. Genus Enterobacterococcus. Epiphysealis durans. These are mesophilic organisms with OTG 37 degrees Celsius. They resist 33 degrees Celsius or 30 minutes. Enterococcus fascicalis. Enterococcus fascicalis. The source of organism is the intestinal tract of human and animal. It is used as a trail starter culture in the manufacture of certain cheese because of its salt tolerance and pH tolerance. It is dominant organism among Enterococcus in India in various milk and milk products. It is non-hemolytic, sometimes may be a hemolytic with graying. Genus Lactococcus. Genus Lactococcus. Lancefield and group grow at 10 degrees Celsius but not 45 degrees Celsius. Reduce the litmus prior coagulating it, growing in broth with 0.1% methylene blue but not in broth with 6.5% NaCl and 9.6 pH. Lactococcus lactis species lactis. Lactococcus species lactis. Elliptical cocci in pair or short chain and elongation of cell in direction of chain. The colony are gray, circular, convex, glistening. OTG is 30 degrees Celsius. It does not produce CO2 or acetyl and does not produce ammonia from arginine. It is an important mesophilic starter. It produces antibiotic and ISIN, which is inhibitory to Bacillus, Clostridium lactobacillus, and some other gram-positive organisms. Some strains produce multi fever due to metabolism of leucine to produce 3-methylbutanol. Lactose is fermented to lactic acid by hemofermentation and produce 0.8% to 1% lactic acid. Growth requirements are very complex. It requires B complex vitamins such as biotin, niacin, thiamine, pantothenic acid, pyridoxine, folic acid, and amino acids such as arginine, valine, histidine, leucine, isoleucine, methionine, phenylalanine, proline, glutamic acid. It is easily inhibited by 0.15 unit of penicillin, 0.5 microorganisms, or oleomycin, and 600 to 1000 ppm quaternary ammonium compound per ml. Lactococcus lactis species lactis bioware diacetyl lactase. Lactococcus lactis species lactic bioware diacetyl lactase. It is similar to the above organism. It utilizes citrate and produces CO2, diacetyl volatile acid, and acetone. Utilization of citrate is plasmin mediated and is unstable character. They produce acetic acid is inhibitory to Pseudomonas coliform Salmonella. Lactobacillus lactis species Cremoris. Lactococcus lactis species Cremoris. It exhibits slow fermentation of lactose. It produces diphlococcine. Genus Pediococcus. Gram-positive non-motile spherical organism, microphilic, showing poor surface growth, occurs as tetrad in pair and short chain. Optimum growth temperature 25 to 30 degrees Celsius. Acid is produced from glucose, galactose, and maltose. Not from mannitol and dextrin. Produced diacetyl appear apparently from oxidation of acetylmethylcarbinol. Genus Leuconostocs. Genus Leuconostoc. Leucos means colorless. Nostoc means encapsulated blue-green algae similar to nostoc except photosynthesis. Gram-positive cocci in pair or short chain. Microaerophilic or aerobic or facultative anaerobic. Heterofermentative. Example production of lactic acid, ethanol, and CO2 from glucose. Some produce slime in sucrose media. It is comparatively inactive in litmus milk and rarely coagulated milk. Growth in medium is enhanced by yeast, tomato, or other vegetable extract. Ferment citric acid to diacetyl acetone to tributylene glycol acetic acid CO2. Cause slit in cheddar cheese made with better culture due to CO2. The organisms are L-mesenteroids species Dextranicum, L-mesenteroid species Mesenteroids, L-paramesenteroids, L-mesenteroid species Cremoris, L-lactis. Endospore forming gram-positive rod and cocci. Endospore forming gram-positive rod and cocci. Endospore forming gram-positive rod and cocci. Genus Bacillus. Bacillus means little stick gram-positive large rod. Aerobic saprophytic soil bacteria may occur in single pair or chain. Aerobic spore former sorts air, water, soil, feed, and water isolated by heating raw milk to 80 degrees Celsius by 10 minutes before planting. Large and rough colonies are formed acid-sensitive, potent spoilage organisms. They may be psychotrope, mesophilic, thermophilic, catalase positive. Spore ellipsoidal to cylindrical in shape. Occasionally bulging sporangia. Bacillus cereus don't grow at 5 degrees Celsius but grow 20 to 35 degrees Celsius. This is important organism because cause food poisoning by producing enterotoxin when the number exceeds 10 की पावर 6 ग्राम. ये तो सेम आ गया है. Group 90 regular known is for forming gram positive rods. Group 90 regular known is for forming gram positive rods. Genus Lactobacillus, leg, milk, bacillus, stab or stick. Long thin rod, gram positive, asparaginous rod, non-motile, microaerophilic, acidic catalase negative, require complex medium like MRS medium, rugosa acetate agar with layering of plates. Source feed, silage manner. 3 group Thermobacterium, Streptobacterium, Beta-bacterium. Thermobacterium group serology E. Lactobacillus, Bacillus delbrueckii species bulgaricus. Thermophilic, OTG 40 degrees Celsius. Used in yogurt, Swiss, Italian cheese. Produce antibiotic bulgaricin, stable for 1 hour at 100 degrees Celsius, which is active against fringe and to a larger extent to S. aureus. Lactobacillus acidophilus. Lactobacillus acidophilus. Thermophilic, OTG 45 degrees Celsius, do not grow at 20 degrees Celsius at 4% salt concentration. Produce lactic acid and acidic residues 1.8%, but to keep viable, the acidic should not exceed 0.6% and 0.7. Produce antibiotic acidophilin, acidolin, lactocidin, medium, asculin, sylvoises agar with incubation at 40 degrees Celsius, 48 hours in CO2 atmosphere. It produces colonies surrounded by dark olive green complex, used along with other mesophilic cultures in kefir, used with beefy bifidum in special ice cream, used in paneer in India, used as capsulated from hypochlorite diet. Therapeutic uses hydrolysis of lactose suitable for lactose intolerant people. Production of antimicrobial substance, antibiotic and H2O2. Prevention of constipation, ulcerative colitis, reduction of cholesterol by fermentation products, destruction of gut microflora after antibiotic treatment because of their capability to grow at low surface tension. Therapeutic value is based on the assumption that this will combat the so-called auto-intoxication caused by accumulation in the body of toxic substance elaborated by toxigenic bacteria. Streptobacterial group. Lactobacillus casei, serology B and C, mesophilic, produce up to 1.5% acid on prolonged incubation. Used in yogurt, it shows inhibit reaction by producing acid peroxidase and antibiotics to Salmonella typhi, Miriam, Legionella dysentery, E. coli, and Originosa. Used along with Candida lipolytica and lactic as cheese flavor additive in processed cheese by spray or freeze drying. Lactobacillus plantarum, serology D, used in brine cheese, OTG 30-35 degrees Celsius. Antibiotic lactolin is produced with which is effective against gram-positive organisms. Beta-bacterium group, Lactobacillus brevis, serology E, mesophilic, OTG 30 degrees Celsius, do not grow at 45 degrees but grow at 15 degrees Celsius. Used in kefir, lactobacillus antibiotic is produced. Genus Listeria. Genus Listeria, gram-positive short rod with rounded and may be cut single short chain or V-form, catalase positive aerobic or facultative anaerobic. Colony are bluish gray when seen by normal illumination and bluish green seen by obligatory transmitted light. Source: whitewater, mud, sewage, pieces of animal and man. Listeria monocytogenes. Listeria monocytogenes. OTG 37 degrees Celsius. Cattle abortion, human food poisoning, meaning accompanied by septicemia. Genus Mycobacterium, gram-positive but staining difficult to high wax content. Acid fast, non-motile, non-spurring, non-branching rod. M. tuberculosis grows very slowly in vitro, requires 2 weeks or more to show visible growth and requires special media for growth. Loifert serum medium cause tuberculosis in human. Group 20. Group 20. Irregular non-spore forming gram-positive rod. Irregular non-spore forming gram-positive rod. Genus Conibacterium. Cell are straight to slight curved rod with tapered and club-shaped form, may appear non-motile asparaginous aerobic or facultative anaerobic, recognized by their banded and beaded clubbed appearance. Metachromatic granules are formed. Chemoorganotropes, OTG 37 degrees Celsius. Seborrheic is not pathogenic and cause rancidity in cream. Pyogenes cause suppurative mastitis. Genus Brevibacterium. Genus Brevibacterium. Rhodcoccus growth gram-positive, non-motile, obligate aerobic, chemoorganotrophic with respiratory metabolism. OTG 20 to 30 degrees Celsius. Non-thermoduric bee illness produce yellow to deep orange red carotenoid pigment, usually present on anterior or surface dependent cheese or Limburger type. Contribute to the surface color of such cheese, add in repairing by proteolysis and improve the flavor and aroma by the production of methanol. Genus Propionibacterium. Propionibacterium, gram-positive, non-motile, asparaginous, anaerobic or aerotolerant. Pleomorphic rod, club-shaped with one end rounded and the other tapered or pointed. Cell may be coccoid, bifid or branched, occur in single, in pair, or short chain in 5 or V and Y configuration. 5% carbon dioxide atmosphere is good for growth. Optimum growth temperature 30 to 32 degrees Celsius. Lactic acid and CHO is converted into propionic or acetic acid plus carbon dioxide. PR Findaranici is associated with cheese flavor because of proline production and I due to carbon dioxide. Physiological grouping. Physiological grouping. Physiological grouping कितना बड़ा है. Physiological grouping. Acid producer capable of fermentation lactose to form lactic acid. Lactic acid coagulates milk by producing precipitation of casein at 4.6 pH. Homofermenter lactose from Lactobacillus. Heterofermenter some Lactobacillus leuconostoc species. Gas producer capable of producing CO2 and H2 from lactose fermentation. Example: E. coli, yeast, Clostridium species. Proteolytic. Degrade milk protein into soluble component by enzyme known as proteinase or proteases. Example: Bacillus species, Pseudomonas species, lipolytic organism capable of attacking milk fed by enzyme known as lipase, liberating glyceride and fatty acid. Example: Pseudomonas species, Archaeobacterium lipolyticum, mold, Geotrichum candidum, Penicillium roqueforti. Sweet curdling, organism capable of causing curdling of milk by the enzyme known as renin-like enzyme before the development of sufficient acidity. X. B. subtilis, B. cereals, and Rococcus liquefaciens propinase, causing change in the viscous of milk or forming threads when the milk is poured from one container to other due to production of gym, gums, mucin, etc. Example: alkaligens, viscous, flavor producing, fruity, Pseudomonas fragile, malty, Lactococcus lactis, maldigens, fleshy, fishy, Proteus, ichthyosis, unclean, E. coli, color fermentation, yellow coloration, Pseudomonas syngenthia, blue coloration, Pseudomonas cyanogens, green coloration, Penicillium roqueforti, black color, Pseudomonas nigrifaciens, red color, Serratia marchanensis. Thank you.